Hello everyone, welcome back to another session of regeneration biology. As I mentioned in the previous class, we split the position control into one basic, that is part A, and then advanced, which is part B. Okay, so position control gene that itself indicates, like so far we have seen there is anterior posterior axis, mediolateral axis, dorso-ventral axis, etc. These are all gradient. We are talking about the gradient, and now how the neoblast responds to them, or how and why a gradient decides or that directs a particular gene expression in a given loci. That means you want eye in the place where eye is there, you want pharynx in the place where pharynx is there. So this is the simple logic during regeneration, and we all know that after an amputation there is blastema that is being formed, which is a highly disorganized or highly rudimentary in terms of differentiation. They are basal cells. So specialized neoblasts are the ones which are produced coarsely in a randomly distributed pattern below the eye, where the eyes are there below the eye or posterior to the eye. There is their distribution. And most important thing you need to know that a eye forming cell, a neoblast that is capable of forming the eye, is present anywhere in the body, or any segment you take that has got an eye specialization, or any segment you take it has got a nephron or a secretory cell producing, or any cell you take it has a neuron producing. So this is the logic. If there are 30 cell types, 30 different neoblasts are present everywhere in any fragment. Means they don't have any biasness. Neoblast specialization can occur regionally. It is spatially coarse. Coarse means mingling or mixed up. That is, specialized neoblasts are distributed much more broadly than the final location. Say final location is eye, it is not located closer to the eye. It can be anywhere in the posterior most portion where the eye is the maximally distant, can also have eye. The logic probably you already got it, because you need to have an eye and head that is being formed even from the segment number 6, like we saw in the previous lecture. Extreme posterior most also should have the ability to form the head and also the eye. So, and this then the final location of their targeted differentiated tissue, where the intended target, final destination, may be far away from the actual position. This doesn't mean that it has to travel from this maximum distance to reach there, because they are there everywhere. It's not that every organ has got only one cell type. It has got many. If an eye progenitor, there are 100, they are present in 100 different spots of the body, rather than they are congregating in one zone. This attribute was first discovered in uninjured animals for eye specialized neoblast, and has important implications for the logic of regeneration. Eye specialized neoblasts are specified between the centrally located pharynx and the eyes, that is the prepharyngeal area. That is the place where they are abundantly located, but they are not restricted there. This is what you should understand. These broadly distributed progenitors ultimately incorporate into uninjured eyes. Why uninjured eye? Eye is constantly being developed because they have to. So none of the eye progenitors are jobless. They are waiting for their turn. When will I get a chance to get into the eye? So this is their way of living. To replace the cells lost due to natural cell or tissue turnover. So they keep on getting incorporated into. So during regeneration, two migratory eye progenitor trails emerge from the wound area. If there is a wound, two trails will come. Let us see what are they. Eye specialized neoblast produce non-dividing progeny cells that progressively migrate to specific target location. They are non-dividing, but their goal is to keep migrating to the target location in the head blastema, where they differentiate. Head is removed, blastema is being formed, and now head structure has to come back, and in that newly formed head structure the eye should form. Not that in that blastoma itself eye is formed. So it is a constant maturation process. Therefore, regulation of migration of the specialized neoblast from the coerce pattern to their target destination appears to be an important aspect of specialized neoblast behavior. So specialized neoblasts are those neoblasts who can give rise to only that particular tissue. They are masters, they are specialized cell types, but they need to know from this coerce pattern, they need to know what is their target, for which the matrix and the position control genes comes into picture. We will see them one by one. So in this picture, as you can see here, there are in the panel A, this picture we have seen before also, where the because they are neoblasts are the only mitotic cells in them, and they are proliferating, and you can see the pharyngeal region where the neoblasts are minimal, and wherever you see the red color dots, they are the ones neoblast population, they are the dividing ones. They are neoblasts are given in red in color, and they are mesenchymal cells distributed broadly. Mesenchyme means that is opposite of epithelium. That is, in cancer you would have studied epithelial to mesenchymal transition. Epithelium means adherent, mesenchymal means roaming around. And the blue color is DAPI stain, that is the nuclear stain, and the pharynx is very clearly visible because it is devoid of, it has the blue color but it is devoid of red color simply because it is minimal in neoblast. And the panel B, that is the transplantation of single neoblast, can regenerate colonies, which we have seen it with broad differentiation potential and capacity to restore regeneration to a lethally irradiated host. This we saw in the previous class, that radiation can restore the animal only if you transplant one. And these resultant animals are naturally, they are genetic clones of the donor. This also we have seen. And they are capable of giving rise to all cell types. In panel C, if you see here, they are specialized neoblast for the eye to produce progenitors that migrate into two different trails from the wound into the head blastoma, from the wound region into the blastoma, because the blastoma is the place where the head and the eye has to restore, where they coalesce into eyes. You can see here in this, this is a cut region, and you can see there are different types of specialized cells are there. The eye forming ones, the so-called eye forming ones, they congregate into, see, this is one eye, this is another eye. They will congregate eventually, and they will have two trails, trail number 1 for the eye number 1, trail number 2 for the eye number 2. So you can see how they migrate through the blastoma into its destined target. And in the D, what you are seeing is the specialized neoblast model, that is the activation of distinct transcription factors in the neoblast specific manner that will seal their fate into the target, and also it will enable the tissue turnover and the regeneration. So no matter whether it is the specialized neoblast migration is part of steady state maintenance of that organ versus it's a newly regenerating structure, the job of the specialized neoblast is fixed no matter what is the situation the animal is going in, and it is governed via the transcriptional machinery. So you can see neoblast, they get into specialized neoblast and they are giving rise to, say, neuron, muscle, or any other excretory cell. So neoblast provide the cellular basis of new tissue production in planaria regeneration in general. So you can see here in this panel, in the E panel, so eyes can be regenerated following a resection, which we know. You can remove eye alone, no need to chop off the whole head. You can simply pluck the eye and you will get back. This is the normal planaria, so eyes are removed and it can restore back its eye. So for this, only eye progenitors need to come into picture. You don't need to have any other progenitor. Eyes can be regenerated following a resection, but this does not trigger the amplification of eye progenitor. Because reason you could imagine, because you don't need to have an increased, in the previous class we saw soon after an amputation, the rate of proliferation of C neoblast increases. But if you just remove the eye, you don't see any accelerated proliferation because the same specialized neoblast can give rise to the eye. You don't need to have any alarming injury response event, etc. You can see the eye progenitors that are magenta in color and they are amplifying. They don't have any increased because you had eye, you simply removed it. They are part and parcel of normal life for the planaria. If I give a simple example, if you have got 1 lakh rupee with you, if 10 rupees lost from that, you won't even notice, although it is a decline. Without too much of effort, that will be restored back. Somehow it will come back, you won't even know. Same logic applies. But if that 1 lakh rupees, if 50,000 is removed, restoring back to 50,000 may need extra earning income, etc. Same logic, the cells existing eye specific specialized neoblasts are good enough to restore the eye, lost eye. You don't need help from other neoblast or no need to have a alarming increase in the proliferation. That is the logic you should understand. In the right side, this can target whenever the eyes are removed, it becomes a blind area, and the target and this targeting can happen even without the eye. Earlier time we said eye will constantly keep replacing just like any other organ, but these cells know where is the eye. But once you remove the eye, how do they know where should they form? Because there is no eye. There is no eye specific cell there. Whole eye is removed. So that thing happens, or that recognition happens mainly because of position control or position specific or the target specific information located. Where and how it is done, we will see in a short while. But some information is there. Even though eye is missing, someone or something tells, oh, here is the eye. So target blind regeneration can happen only because of this information. Regeneration occurs as an emergent property of constant progenitor production. During regeneration, with no progenitor production rate change, if you remove just the eye, there are fewer cells in the forming eye accessible to undergo cell death, and they are young. Consequently, what happens? Fewer cell death events per eye passively enable regeneration. Normal eye, what happens? Let us assume one eye has got 100 cells. 3 cells die every day. So 3 cells need to be replaced. So it is a constant procedure. When eye is missing, what happens? 3 cells came in, they don't die because there was no cell to die. 3 cells just came. And one more 3 cells will come next day, it became 6 cells. One more cell came, it became 9 cells. They are not dying because these are all young cells. In the other case, some are oldest one and they are dying. So these are relatively newer cells, hence death is not there. It's just like pouring water into a vessel which has got a hole in the bottom versus which has no hole. So normal, if the rate of water flow into the pot is same as the hole, any given time water will be filled. This is a scenario in a normal eye. In the absence of water and absence of hole, any water falling in will stay contained inside. And once it reaches a threshold, it will start, you can consider to that of overflowing. Okay, instead of hole, it is best is to think as an overflowing. 1 liter pot, if water is falling, if it is overflowing, they will equalize out. But if pot is empty, same as eye is removed, any water falling in will continue until the pot is filled before it can overflow. So same way, they will keep migrating and they keep filling and the eye will be formed completely. So this will consequently fewer cell death events per eye passively enable the regeneration. Eye formation after amputation. So decapitation also gets rid of the eye. If you remove the head, you did not do anything to the eye, but the whole area is missing. Okay, following decapitation, eye progenitors are amplified in large numbers naturally because eye is missing. Head is missing, naturally eye is missing. And they can bring rapid eye regeneration in the new head. New head is formed from the blastema, and then the new eye also has to form, and naturally there is rapid proliferation of neoblast along with the C neoblast. along with the eye-specific neoblast or specialized neoblast. Would the absence of only the eye following eye-specific resection lead to similar eye progenitor amplification? And it will not. What about removal of only few cells? Would the amplification of only few progenitors occur? You did not remove the whole eye, you removed maybe 25% of the eye. What will happen? The surprising answer is that specific removal of the eye leads to no change in the number of eye progenitors, but the eye will regenerate nonetheless. Because the reason I told you, maintaining an eye is a routine process. Maintaining, how it is maintaining? It is removing cell, adding new cell, removing cell, adding new cell. So hence you remove little bit of cell or half of the eye or a full eye, getting a new eye do not require any active involvement of or more progenitor proliferation. You don't need to increase the production of progenitors. It's like a 100-member family. If 3-member visitors come, they don't need to cook extra. 100-member family, 3 extra people came, they don't need to cook extra. But if it's a 3-member family, even 1 person came or 2 person came, it is a huge task. This logic you should keep in mind because the eye-specific progenitors are present throughout the body. All you removed is one eye and the progenitor number is so high that it can make multiple eyes. And don't make multiple eyes everywhere, but it's constantly replaced. Same logic applies to other. Eye is a specialized organ which you can easily spot it and very easy to monitor it under a microscope. Unlike other cell type. That is why people work on eye, but other cells also follow or other tissue types also follow the similar pattern. In animals with one uninjured eye and one regenerating eye. Think about it has 2 eyes. You removed only one. Now that is an interesting scenario. You removed regenerating eye because you removed one eye. The 2 eyes incorporate progenitors at the same low rate. So let us assume 3 cell or 5 cell or 10 cell. 3 cell I gave it for an example. So one eye intact, one eye you removed. So both eye, not that the removed area gets 100 cell per day. No, both eye is getting 10 eye per day. But in the existing eye, 10 cells die also. Whereas in the missing eye, 10 cells, first day, nothing is there to move out. One more 10, then become 20 cells. No more cells are moving out. So whatever is coming in, the same logic of one mug of water you are filling in. Until it is full, there is no way it can overflow. Yet one eye will grow. So this is the logic. Incorporate the progenitor at the same low rate, yet one eye will grow. And other eye will stay constant size. How? Answer I already told you. Eye size is determined not only by the rate of new cells entering an eye, but also the rate of cells leaving the eye through cell death. Leaving means they are not traveling, they are just being killed off or shedding off. Thus, if the rate of new cell incorporation into a regenerating eye is not changing, then the rate of cell loss in the regenerating eye per eye should be less than the loss rate of an uninjured eye. Uninjured eye has got many old cells. Newly forming eye has got every cell is young. Only their difference is by 1 day. Whereas the existing eye may be few months old. The instructions for tissue turnover and regeneration, how it's working. So tissue turnover is one thing where it has to constantly supplying new cells. Constitutive positional information. Positional information is very important. Constitutive means any given time this information knows, yes, this area is for eye, this area is for neuron, this area is for brain, this area is for pharynx, like that. Beta-catenin RNAi. Again, we are going back to beta-catenin because lots of research has been done or lots of this anterior-posterior axis is maintained via Wnt beta-catenin signaling. Beta-catenin RNAi led to dramatic regeneration outcomes. Heads were regenerated in place of tails. You can make them if you block. We have seen it. If you make cut and head will normally form, but if you block Wnt signaling, beta-catenin RNA, the other end also you will end up getting a head, resulting in 2-headed animals with both heads fighting to pull the same body in different direction. That is a very funny situation where every animal move where its head is there. So it will move towards that direction where the animal's head is there. You can look into any worm or any caterpillar, any worms, tiny to small, or a snake, anything, wherever its head is there, no animal will keep its head in the extreme backside and then travel. No one moves backward. Of course, some animals transiently will do that, but default movement is forward. But if planaria has got two heads on either side, it will be stretching the head on either direction. So any given time it will be moving around and rotating around. That means it's not simple thing. That shows both head have got equal weightage. That means fully functional. It's not a dummy head. That's what this logic says. This observation suggested a role for wind signaling in the decision making process at the transverse amputation planes to regenerate a head or tail. So you can give an added value to the wind signaling, that head formation is important for a decision making process of the newly formed animal. So you can tell wind signaling was important for deciding which direction the animal will go. It's a decision, right? So you want to make just like you are designing a bike, you are thinking whether where should I keep the handle? Should I keep the handle in front or back? It's purely based on the engineer. So here the wind signaling decides where the head should form. Accordingly, wind signaling decides where the animal should form, because the head decides that. Following beta-catenin RNAi in uninjured animals, a slow and steady transformation of the body plan occurs during tissue turnover. Many heads appear around the animal's periphery. If you block, no injury, nothing, took a planaria, blocked wind signaling. Wind signaling, you just gave shRNA against beta-catenin. What will happen? Heads will start forming everywhere. Means any area is vulnerable to form a head. That means the position of, or the overall position, location of a structure called head is decided by one signaling event, or the positional information is so important that usually that we saw in the previous class also, in a non-regenerating species, after decapitation, if you block wind signaling, you can end up getting a head. So now in a normal animal, if you block wind signaling, you can end up getting multiple heads. You can imagine how plastic the system is. So wind signaling upregulation through APC or NOTAM. NOTAM is a carboxyl esterase that removes an essential palmitoid moiety from winds to deliberated. So basically it's a functional role, and it forms the first non-extracellular protein deacylase. It's not important to know, mechanism is written down here, but APC is a negative regulator of beta-catenin, means beta-catenin is degraded if APC is active. If APC is functionless or jobless, beta-catenin gets stabilized. So wind signaling can be upregulated if APC is removed. Can result in the regeneration of tails in the place of heads. The opposite. When wind signaling is inhibited, you get heads, but you favor wind signaling by getting rid of the negative regulators of wind signaling, you will end up getting the opposite, that is the tail. That is, the polarity is maintained by wind signaling. These findings indicate that constitutive regional expression of wind ligands, that is usually posteriorly, and wind inhibitors anteriorly. Near the head region, wind inhibitors rule. But the tail region, the wind activators rule. Control regionalization of the planarian apiaxis. This positional information decides how different tissue structure is formed and how it is functioning. And now comes the dorso-ventral axis. Dorso-ventral axis is something which is very interesting, and dorsal means your backside, ventral is your belly, your chest region, that is ventral. Another prominent model of planarian adult positional information involves the BMP signaling. We kind of discussed about it in the previous class, which regulates the dorso-ventral axis. BMP inhibition, if you inhibit the BMP, there are various methods. You can use RNAi, etc. Because the good thing with planaria is that you can feed the animal with an RNAi. Normally it goes in, since it's a small RNA, it gets into the circulation and it inhibits. So it's a very easy system to work with. So BMP inhibition results in progressive ventralization. What is ventralization? Let us assume you know your chest. Normally you have got your umbilical cord, your back and your front are different. They are not the same. Now if you block the BMP signaling, in planaria ventral side there is a pharynx also is there, that is coming from the ventral side. Now if you block the BMP signal, you end up getting ventral side on either side. Just like you saw head on both sides, just like you can make a tail on both sides through tweaking the wind signaling. If you block BMP signaling, you will get ventral side on either side. And which includes, there are real solid structures are with ventral tissue types are predominant, such as nerve coats, ciliated epidermis appearing dorsally, which are supposed to be in the ventral region. Now it is gone to the dorsal region. It is just like we all say, what is east? Sun rises in the east. If sun rises from the west or from the north, will you call sun is rising from the north, or will you call north as east? So this is a conundrum. So if you inhibit BMP signaling, you end up getting these structures, ventral structures appearing in the dorsal. So if your legs are coming from your shoulder, you can say that you got posteriorized. Means leg is a posterior structure. Hand is an anterior structure. Hand came on your legs. We can call the person got anteriorized. The anterior structure went into the posterior. Same way vice versa. Posterior structure came into the anterior region, we can call you got posteriorized. Same way it got ventralized. As the so-called dorsal region, you have got ventral structure. For example, BMP4 RNA. BMP is a ligand of the BMP signaling. RNA animals can flip over and glide on their back. They're gliding on their back naturally because the animal think their back is their belly, chest. So instead of moving with the belly down and back up, it will go with the back down and belly up. So this is decided by the BMP signaling. DV axis is also governed by roles for other pathway regulator. Dorso-ventral axis, that is DV axis, including ADAM, that is a variety of noggin, that is also known as NOG and noggin-like genes, which are antagonist to the BMP signaling. Just like we were said Wnt activators and Wnt inhibitors, like BMP signaling activators and BMP inhibitors. The BMP4 is expressed dorsally, naturally, because you want BMP signaling, and in a medial to lateral ML gradient. So dorsally, backside, in a medial to lateral. That means your chest to the right hand side, chest to the left hand side. So chest is your central portion to the right hand side, central portion to the left hand side. That side of the dorsal side, that is BMP4's place where it is expressed. Now on the ML axis, the Wnt5 mediolateral axis, the Wnt5 is expressed laterally and inhibits the medially expressed slit, another gene called slit. Wnt5 RNAi and slit RNAi results in medial lateral patterning abnormalities. So these are all interplayers like slit, noggin. These are all influencers of BMP signaling. And what is important to understand, Wnt5 RNAi and slit RNAi result in medial lateral patterning abnormalities. Or in other words, it is difficult to explain. Say your right hand now came on the left hand side. You know how your right palm looks like. What if it came on the left side? Your thumb, instead of looking towards your right hand thumb looking towards your chest region, now your right hand thumb will look away from your body. Isn't it? Usually this is the variation you will get it. That is the mediolateral patterning also is to be followed, not just a dorsoventral patterning. Genes with constitutive and regional expression and the association with a patterning are called position control gene. In short form, we call them as PCGs. We will study more about these genes and position control in the next class. Thank you.